Welcome back, everybody. We are rounding up the semester. We have 2 labs left, brain and special senses. Today, we're going to make you smarter by studying our brains. One thing I wanted to mention before we get going is that in infants, our brains are making millions of new synapses per second. That means they're building new connections with other neurons every second. As we get older, this number of synapses declines very quickly. Neurons that fire together, wire together is the saying. So the more you're using these pathways, the more we learn, the more you walk and fall, the more you are practicing anatomy or an instrument or maybe a sport, the more you're wiring these neurons together. And so those connections get stronger and the other ones get weaker. So as we get older in childhood and adolescence, our brains are sort of casting aside the synapses that we don't really need anymore. And I like to think of this as like sculpting a statue. We're chiseling away at this block of marble, getting rid of stuff that we don't need. In labs, you'll be looking through microscopes at nervous tissue. Here we can see a neuron, this dark staining neuron. This is the cell body or the soma, and we can see the nucleus right inside of that. These smaller little dots or nuclei here are glial cells. They're supporting cells with a wide array of functions that you'll be learning more about in lecture. There's actually a lot more glial cells than there are actual neurons. Neurons have structures called dendrites. The dendrites are receiving electrical signals from other neurons. The signal travels down the dendrite to the soma, and from the soma, it travels down the axon. These electrical signals are only traveling in one direction. So the function of our nervous tissue is to do just that. Transmit electrical signals that help us learn or process information, either sensory information coming in or sending out motor commands to the body. In our central nervous system, we find this in our brain and spinal cord. In our peripheral nervous system, we find this in our spinal nerves. A couple of directional terms and landmarks we're going to need to know is that one, our brain and spinal cord are part of the central nervous system. Just like we learned about our axial skeleton, the central nervous system runs down the center of our bodies. We have 2 terms that are usually used with animals, rostral or towards the nose, so this would be anterior, and caudal, which means toward the tail or posterior. The major parts of the brain are this really large structure here called the cerebrum, and that makes up about 80 to 85% of our brain volume. Back here we have the little brain, also called the cerebellum, and that has about 50% of neurons. We have our brainstem here, and that brainstem then will turn into our spinal cord. The central nervous system, the brain and the spinal cord, are surrounded by meninges. Meninges are connective tissue membranes that are going to offer protection to the central nervous system. If we look at this image, we can see that the most superficial layer here is our skull or our bone. And deep to that, we have our first layer called the dura mater, M-A-T-E-R. So not matter, but mater. This basically means tough mother. Our outermost layer is the periosteal layer. Peri means around or near, osteal is bone. So this is the layer that's around or near the bone. And we can see that right here. Deep to that is our meningeal layer. Now our next layer deep is our arachnoid mater, or the spidery mother, because there's these little spider-like projections that stick down into our subarachnoid space. And in the subarachnoid space is where we'll find cerebral spinal fluid, or CSF. The third layer is called the pia mater. When you do your brain dissections this week in lab, you'll see this shiny, almost like a saran wrap covering that's tightly adhered to the brain. Other structures that you'll need to know and that you'll see in your dissections as well as in the models of the brains are the ventricles. The ventricles are going to be producing cerebral spinal fluid. From an anterior view, you can see we have a lateral ventricle on the right and a lateral ventricle on the left. These converge on the third ventricle, which then leads through the cerebral aqueduct into our fourth ventricle, and that will lead to our central canal, our lateral and median apertures. What does cerebral spinal fluid do for us? Well, it's filling the ventricles and our canals, and it's essentially a bath for our brain. It's helping to clean and wash away toxins from around our brain. We make about 500 milliliters of this per day. It also adds buoyancy and helps our brain sort of float inside of our skulls and offers protection and cushioning from hitting the inside of the skull. What we'll see is that it's draining from that fourth ventricle and surrounding our brain and spinal cord. Let's look at this pathway here. If we start at number 1, right here, this is in our lateral ventricle, and we have a structure called the choroid plexus. In your brain dissections in lab, when you take a mid-sagittal cut through the brain, you'll be able to take a probe and put that probe into that lateral ventricle, and if you look carefully, you'll see some blood vessels and a little pinkish structure, and that's your choroid plexus. This choroid plexus is going to produce cerebral spinal fluid that fills the lateral ventricle, and it's going to move through the interventricular foramina into our third ventricle, where you'll see more choroid plexus that's going to add to that cerebral spinal fluid. The CSF then passes down through our cerebral aqueduct into our fourth ventricle, where we have another choroid plexus that's going to produce more cerebral spinal fluid. And that fluid then leaks out through the median and lateral apertures and also passes down into the central canal of our spinal cord. So now that cerebral spinal fluid essentially bathes our brain as well as our spinal cord. It's then taken back into circulation through the arachnoid villus. One condition called hydrocephaly basically means water head. This is the swelling of the head due to the overproduction of cerebral spinal fluid. This fluid accumulates in the ventricles and puts pressure on the brain. That brain, as it swells, can get damaged by pushing on the skull, and this can lead to a lot of complications and mental disabilities. Physicians can take a shunt, put that shunt into the ventricular system, and run that into a blood vessel to divert that cerebral spinal fluid into the bloodstream. Let's pause here, take a few moments and review some of the terminology that we just went over. See you in a few minutes. Our brain, as you can see, is separated into a right and left hemisphere by this longitudinal fissure. You might notice that on this brain, we see extensive folds. These folds are called gyri, a single being a gyrus. And in between those folds are grooves called sulci, a single groove being a sulcus. Now this is going to increase surface area in our brains so that we have more room for neurons. If you were to take a cross-section through our brain, a coronal cross-section through the brain, what you would notice is that we would have about 3 millimeters of gray matter on the outside, unmyelinated neuronal tissue, and white matter on the inside. This is myelinated nervous tissue. If we look at the cerebrum a little bit closer, we'll notice that the cerebrum is divided into different lobes. And if you remember, some of these terms should be familiar from our skull lab. So we have our frontal lobe here in blue, the parietal lobe here in purple, temporal lobe in yellow, and the occipital lobe in green. The frontal and parietal lobes are separated by the central sulcus. Anteriorly, we have the precentral gyrus, which is part of the frontal lobe, and posteriorly, we have the postcentral gyrus, which is part of the parietal lobe. Down here, we can see the cerebellum, or our little brain, and we'll see that the temporal lobe is separated from the frontal and parietal by that lateral sulcus. In general, we can say that the frontal lobe is responsible for our voluntary motor functions, planning, our general mood, and our social judgment. Our parietal lobe is responsible for bringing in sensory information and integrating a lot of that sensory information. Our occipital lobe is responsible for our visual information, or processing our visual sensory information. And our temporal lobe is receiving information about hearing, smell, learning, memory, emotional behavior, and taste. Now, it's important to note that these lobes are communicating with each other. They're communicating with the cerebellum, the pons, the medulla oblongata. They're also communicating between hemispheres. So there's a lot of information processing going on here. Taking a closer look at each lobe, we can say that the frontal lobe, again, is responsible for higher thought functioning. Now, part of that frontal lobe is a region called Broca's area. And Broca's area is responsible for our motor speech. So if that area is damaged, we have trouble speaking because we're having an issue transmitting information to the muscles that are going to help us say the words that we want to say. I'd recommend you take a few minutes to click on this hyperlink. It'll take you to a video on YouTube of a girl who is 19. She had a stroke and she's being interviewed. And you can see the struggle that she has communicating and speaking the words that she's trying to say. We mentioned before that the central sulcus is that posterior border, and our precentral gyrus here is our primary motor cortex. So a lot of our motor signals are going to be sent out from this precentral gyrus. This is an image called a homunculus. On the left, we can see our primary motor area, and on the right, we can see our primary sensory area. The primary motor area is associated with our precentral gyrus here, as you can see in red. And what this is representing, or what this is showing you, is how much of this precentral gyrus is dedicated to controlling muscles of our face, lips, jaw, as well as our hands, fingers, thumb, wrist. And if you think about our fine motor skills, there should be a lot of control to our hands, wrists, fingers, and the communication that we have with our faces. Now in the bottom left, this is just another image or a model that somebody made to show that motor control over these different parts of our body. Now there was a guy in 1848 named Phineas Gage. He was a foreman on the railroad, and he was packing blasting powder into a hole. Great idea, right? Using a metal tampering rod to pack blasting powder into rock. Well, at some point, that metal tamping rod caused a spark, ignited the blasting powder, and it launched his tampering rod up through his face, right around his zygomatic bone, behind his eye, and out through his frontal lobe and frontal bone. Now, Phineas Gage was said to be a very nice, congenial person, really responsible in his life. But after this accident, his personality was very volatile. And so he was very different than what happened before. And so people, first of all, were totally amazed that he even survived, because a lot of the thinking was that if you damaged your brain, you would just die. But second, they were really curious as to why his personality changed after this accident. Because of this accident, Scientists started to explore schizophrenia, depression, and different anxiety disorders, and trying to figure out what was going on in that frontal lobe of the brain. One scientist named Walter Freeman spent about 40 years of his career doing lobotomies. Basically, he would take this metal rod and he would go into the superior orbit, hammer in that rod, and basically mush up the frontal lobe. And he thought that this would help cure various mental diseases. I would recommend clicking on this link if you have about 45 minutes to an hour and watch this documentary about him. It's really interesting. The parietal lobe has our postcentral gyrus, and this is our primary somatosensory cortex. You'll also see an area called Wernicke's area. It overlaps with the temporal lobe. Wernicke's area is involved in language formation. So we have the motor control to speak the language, but we have a hard time actually forming the language. So if you click on this link, you'll see another YouTube video of an older woman trying to develop language, but unable to communicate anything to the person who's interviewing her. So back to that homunculus. If we look over here on the right, this is our sensory area. Again, a lot of sensory information related to our tongue, our face, our lips, and as well as our hands, fingers, and thumb. So we can see this model down here showing how much sensory information is coming in from these areas to the postcentral gyrus. Our occipital lobe is responsible for visual information. If you've ever been hit in the back of your head, you may have seen stars, or if you kind of thump your fist against the back of your head a little bit, you might see that your vision sort of bounces around a little bit. There is the transverse sulcus back here that's separating the occipital lobe from our cerebellum. The temporal lobe is responsible for processing our hearing, our smell, and our taste. So auditory, olfaction, and gustation. So pause your videos, practice the information related to our lobes, and we'll move on here talking about other parts of our brain. Welcome back. I mentioned earlier that our brains are divided into right and left hemispheres by that longitudinal fissure. Now, you've probably heard people reference this. I'm more left-brained, I'm more right-brained. And generally, our left brain is involved more with facts, math and science, detail-oriented and logic, where our right brain is more involved in imagination or symbols, music, and art. I'd recommend checking out this podcast here by Hidden Brain, One Head, Two Brains. It goes into the detail about the differences between the two hemispheres, but also how the two hemispheres will take over for the other hemisphere if there is some kind of brain damage. There's a structure that you'll see in labs during your dissection that you'll cut through called the corpus callosum, and just under here you'll see the lateral ventricles. The corpus callosum connects the two hemispheres. And again, down here, I have another podcast that I think is really cool that explores a person who had their corpus callosum cut to reduce epileptic seizures. Inferior to that corpus callosum, we can see our thalamus. And our thalamus is the relay center of our brain. It is taking in a lot of sensory information and figuring out where that information should be sent. So is it going into our parietal lobe or into our temporal lobe? It's also helping to integrate that information that's coming in. Below that is a region called the hypothalamus. And the hypothalamus is an endocrine gland, and it works with the pituitary gland. It controls the pituitary gland. It sends hormones that cause the pituitary gland to release or stop releasing other hormones. It's involved in our autonomic nervous system, our thermoregulation or managing our heat, food and water, sleep, and memory. Posteriorly, we see the epithalamus, and there is a gland called the pineal gland associated with the epithalamus. The pineal gland is involved in our circadian rhythms. When the sun goes down, it gets dark outside, the pineal gland produces melatonin, and that makes us sleepy. There's a disorder called seasonal affective disorder that people in higher latitudes generally experience. There's almost no sun in the winter, and so they get depressed. Moving a little more inferiorly, we have our midbrain here, and our midbrain has our cerebral aqueduct that travels between the third and fourth ventricle, and a structure here called the corpora quadrigemina. On top, we have the superior colliculi, and on bottom, we have the inferior colliculi. During your brain dissections, if you take your sheep brain and you grab the cerebellum and you pull down and you take the cerebrum and then you pull up, you should be able to see these 4 little nuclei. In the hindbrain, we have our cerebellum, our little brain. And again, this is divided into hemispheres separated by the vermis. Now the folds here are called folia instead of gyri, but essentially they're the same thing. The cerebellum is responsible for muscle coordination, motor skills, and also involved in time. In a mid-sagittal cross-section of your cerebellum, which you'll do today, you'll see that the cerebellum has this white matter, and it's also called the arbor vitae, or the tree of life, which, when you think about the folds being called folia or foliage or leaves, kind of makes more sense. And that sits right up here on top of that fourth ventricle. Our pons, a little bit more inferior here, is involved in relaying information about movement and sensation from that cerebrum back to the cerebellum, and it's involved with respiration, sleep, and taste. Our medulla oblongata is a part of the brainstem. It regulates our autonomic nervous system, but it's also involved in our heart rate, blood pressure, and respiration. Because most of you are living on campus and there's oftentimes social pressure to get involved with alcohol and drugs, I wanted to put in a little bit of information about how alcohol and drugs affects our brain. Severe alcohol consumption can damage our cerebellum, which we mentioned is involved in muscle coordination and balance. THC, the active chemical in marijuana, can affect our prefrontal cortex as well as our temporal lobes and affect our attention and memory and motivation. Cocaine can actually affect up to 90 different parts of our brain and damage blood vessels in the brain. It also alters and changes our levels of neurotransmitters like dopamine and serotonin, which can cause mood swings. Ecstasy damages nerve cells that respond to serotonin, and so again, this can cause mood swings, but it also affects our thinking and our memory. And finally, LSD. LSD is going to affect the cerebral cortex, so it's going to change the way we're thinking, the way we're perceiving the world, and also the locus coeruleus. And this is a structure that's involved with sensory signals. So LSD allows more sensory signals to be brought in, where a lot of that sensory information actually gets filtered out. So we're perceiving a lot more sensory stimulation and seeing brighter colors or hearing sounds or seeing things morph and change shape. So go ahead, practice this for a little bit, and then we'll move on with cranial nerves. Welcome back. Let's talk about our cranial nerves and finish up with our brain lecture today. We have 12 pairs of cranial nerves coming off of our brain that are going through those foramina and those fissures that you learned way back when we looked at the skulls. First, we have our olfactory nerve, and I also wanted to point out that you'll see these letters in parentheses. This is Roman numerals. Some people might not be familiar with Roman numerals, but the I is 1, 2 I's is 2, 3 is 3. When you see an I before a V, you subtract. So a V is 5. So IV is 4, VI is 6, And then we move down here. X is 10. So we can see the X is 10 here. Oh, I before an X, that's 9. And then 10, 11, 12. So our first one is the olfactory nerve. 2 is our optic. 3 is our oculomotor nerve. 4 is our trochlear. 5 is our trigeminal. 6 is abducens. 7 facial. 8 vestibulocochlear. 9 glossopharyngeal. 10 vagus. 11 accessory. And 12 hypoglossal. Some of these you'll be able to see on your brain dissections, but I would spend a lot of time studying this diagram. You're going to need to know the functions of these, as well as what foramen and fissures they're passing through. As always, we have a mnemonic for you. On occasion, our trusty truck acts funny. Very good vehicle anyhow. You'll also need to know which of these cranial nerves are sensory, which are motor, and which are both. Sensory nerves are bringing information from organs or about the external environment from receptors back to the brain to be processed and integrated. Motor nerves are taking commands from our integration centers out to our muscles or glands or organs to respond to the sensory information. Some of these nerves have both functions, sensory and motor. So you can use the mnemonic. Some say marry money, but my brother says big brains matter more. We're going to run through a handful of slides, 12 slides, looking at each of these cranial nerves. You might see on a test an image from this PowerPoint asking you about that nerve or the foramen or fissure it's passing through. You might see a brain dissection on a test asking you about a specific nerve. You'll see models that are out, and you might even see a skull with a pipe cleaner sticking through a foramen or a fissure, asking you to name which nerve is passing through that. Run through those mnemonics a couple times. It'll help you to be familiar with the names of these nerves as we work through these next 12 slides. Welcome back. So our first nerve is the olfactory nerve, or cranial nerve number 1. If you remember the cribriform plate of our ethmoid bone, we'll see that the olfactory bulb is right here, just superior to that bone, and we have these fascicles coming down into the nasal mucosa. Next lab, we'll see how this works. This nerve is responsible for olfaction and sending sensory information back to the brain about what it is we're smelling. The optic nerve is cranial nerve 2, and it's taking visual sensory information from our eyes and sending that back to our occipital lobes. This nerve passes through the optic canal in that orbit. Our oculomotor nerve, cranial nerve 3, is responsible for eye movement, and oculomotor should help you out with that. It's passing through our superior orbital fissure. The trochlear nerve, also passing through the superior orbital fissure, facilitates eye movement. Cranial nerve 5 is a little more complex. This is called the trigeminal nerve. There's 3 branches. Our first branch is the ophthalmic division, V1. And down here in the bottom left, you can see the 3 areas that are innervated by the 3 branches of the trigeminal nerve. So our ophthalmic division is a little bit of our nose, our scalp, and our eyes. That ophthalmic division, if you follow this down, is passing through that superior orbital fissure, bringing information from our nose and our eyes and our forehead. Our maxillary division, V2 here, if you follow this along, is branching off of our trigeminal nerve, passing through the foramen rotundum and innervating our maxillary region and our teeth. The mandibular division, V3, is right over here, passing down through that foramen ovale to our mandible and muscles of mastication. So our lateral pterygoid, medial pterygoid, temporalis, and masseter, which we learned about before. Cranial nerve 6 is our abducens nerve, and that's also passing through that superior orbital fissure. Out of our first 6 cranial nerves, 3 of them facilitate eye movement, and 1 of them is bringing in sensory information from our eyes back to our brain. Cranial nerve 7 is our facial nerve, and so our facial nerve is passing through that internal auditory meatus we learned about. And this is going to help us with facial expression. It's going to send sensory information about taste from the anterior 2/3 of our tongue. It's going to help control salivary glands, tear ducts, as well as our nasal and palatine glands. Our vestibulocochlear nerve is also passing through that internal auditory meatus. You'll see that there is a vestibular nerve, and we'll learn more about what the vestibule is next week, and a cochlear nerve that are then merging into the vestibulocochlear nerve. This is going to provide information or bring in sensory information about hearing and balance. Next is our glossopharyngeal nerve. This is passing through our jugular foramen. It's providing control over swallowing, salivation, gagging, and it's sending sensory information from the posterior 1/3 of our tongue, as well as controlling blood pressure and respiration. Our vagus nerve, passing through that jugular foramen, controls swallowing, speech, and regulates a lot of our viscera in our bodies through the parasympathetic nervous system. Our accessory nerve, number 11 here, is passing through our jugular foramen, and again is providing information over swallowing, as well as our head, neck, and shoulder movement. And we can see it innervating our trapezius and our sternocleidomastoid muscles. And last is our hypoglossal nerve. This is also providing motor control over our tongue for speech, food manipulation, and swallowing. Out of those last 6 nerves, 4 of them are involved in controlling swallowing. The first 6 nerves, 3 of them were involved in motor controls of our eyes. So on a test, if you're trying to identify the function of a nerve and you can't remember or you're blanking, chances are if you throw out eye movement or swallowing, you might get that question correct. In labs this week, you're going to be dissecting a sheep brain. You'll get more extensive instructions from your lab TAs, but please do communicate with them if you have any cultural or religious issues doing a brain dissection, and we have all kinds of supplemental materials to help you be successful. So spend some time here, practice all of the information we just went over. Good luck on your tests. See you next week for our final lab.